This episode is supported by two companies deeply invested in advancing GPCR research, Orphans DiscoverX and GeneTex. Orphans DiscoverX helps GPCR teams turn signaling insight into defensible data with validated assay platforms that scale from hit ranking to regulatory submission. And GeneTex is on a mission to make GPCR antibody reliability something the field can really count on. Rigorous validation, community collaboration, and free samples available for you researchers, ready to put them to the test. Links to both are in the show notes. And now, let's dive into this episode.
So hello everyone, this is Yamina from Dr. GPCR, and today I'm very excited to have with me Charlotte Crauwels. She is finishing up her PhD, and Charlotte and I have been trying to get and sit down and have a chat for a very long time, and I'm excited that we finally got to do it. Charlotte, welcome to the podcast.
Thank you for having me.
I'm very excited to have you on. So why don't we start by you introducing yourself, telling us who you are and what is it that you're working towards right now?
Right. So as you just mentioned, I'm a final year PhD student here at the Free University of Brussels in Belgium. I'm working in the group of Wim Franken. Are you still there?
Yes, I am. Can you see me?
Oh, no, that's why I can see you. Is that supposed to be like that?
You can. I'm here.
Yeah.
So it says videos in the room may appear lower quality than the recording, which are captured at full quality. So I see you. You're a little bit blurry, but I can still see you.
Oh, okay. Perfect. Sorry about that. I thought you disconnected, that there was something with the Wi-Fi.
No, I'm here.
Okay. Well, I guess I will start again.
Start over.
Yes. So as you just mentioned, I'm indeed a final year PhD student here at the Free University of Brussels in Belgium. I'm working in the group of Wim Franken, and I think I would define myself as a computational protein designer that is focusing on GPCRs. And I'm very excited about this podcast. So let's get this started.
Very excited to have you on. Since you're finishing up your PhD and this is your last year, I'd be curious to hear about how you, looking back at the PhD and at the trajectory that got you here, what drew you to the work that you're doing right now?
Well, that's a long story. It was definitely not very, I'd say, linear. I wasn't planning doing a PhD. I wasn't planning on working on GPCRs, but it all went, I don't know, step by step. I ended up here. I was actually studying bioengineering here in Brussels. And during my master's, I got introduced to bioinformatics. I liked it a lot. I feel quite comfortable behind my computer. With the feeling that I have all this data on the tip of my fingers. I think it's pretty cool. And so I decided to do my master thesis in bioinformatics. I was studying antibodies and nanobodies and how they bind to their antigens and so on. And I think the master thesis went pretty well. I enjoyed doing research. My supervisors at the time seemed to be quite happy about my work, and they actually encouraged me to do a PhD. So I thought, okay, maybe why not? Because I was actually also not ready, I think, at that point to start working in a company. I didn't know what I really wanted. So I thought, okay, maybe a PhD is a solution. It would I would still be able to explore, and then maybe by the time I finish my PhD, I will know what I want and what I really like to do and to work on. So I started sending some emails to all or almost all computational research groups here in Belgium. Just saying that I'm looking to do a PhD. I'm not sure what I would like to work on. I just want it to be computational work, a rather applied topic, and also that I'm driven by engineering life. I like to think that now we have all this data available about like the biological systems around us. We have quite some knowledge about these systems, and we have quite a lot of powerful tools to study these systems. So I think it's the time to use these tools to start playing with the systems to then maybe optimize them or gain insights into them. So with that in mind, I contacted these professors, and then one thing led to another. I got in contact with Wim Franken, my current supervisor, and he said, well, I know this this spin-off of our university that we maybe could collaborate with, so that you can do applied research. And so that company is called Therapeutics, and they work on GPCRs.
I think you've muted yourself accidentally.
Oh, is it okay like this?
Yeah. Okay. So I got to a point where you said that Therapeutics and they work on GPCRs.
Exactly. So we got in contact with Therapeutics. They work on GPCRs. We started exchanging some project ideas, what we could do together. And then one thing also, like, again, led to another for some administrative reasons. At the end, I could not work or collaborate directly with them because I got a funding, personal funding, which does not allow you to work just one-on-one with a company. And so now I'm actually not related to the company anymore, but I still kept the GPCR related projects that we started developing together. And so here I am now in my final year, still working on GPCRs, and I'm actually very happy that by coincidence I happen to be studying them.
If you were not a computational protein designer, what else would you be doing?
When I was a kid, I always wanted to become a doctor.
It's a different.
Yeah, okay. I think many of us actually. I think many of us actually want to become a medical doctor when we were kids. I think the idea that you could like fix people or save them is like quite exciting, and the idea that you could have like impact, real impact, or at least that's how you see it when you're a kid. And but then just before starting actually enrolling for med school, I actually realized I cannot stand the sight of blood. I feel like I'm gonna faint if I do. So I right before starting uni, I said, okay, maybe another good option is to start bioengineering studies. I think they have a very broad curriculum, and I thought again, like I'm not sure what I want to do, so let's do that and then figure it along the way. So I was definitely not planning on becoming a researcher, but when I think back about it now, I think it was actually maybe I could have foreseen this, because I was always the one in the classroom that would be like asking a lot of questions after the lectures, and I would be like, okay, but what if, or what's next? And now as a researcher, I can actually ask daily myself the questions, what's next and what if, and I can go try to find these answers myself, and I think it's super exciting. So again, it was not planned, but actually step by step I became a researcher, and I'm super excited about that.
A couple of things worth mentioning before we continue. The Dr. GPCR University exists for those of you who want training that keeps pace with how the field actually evolves. Expert-led, live and on demand, and built to serve discovery. And Terry's Corner with Dr. Terry Kanakin is where the frameworks that matter in drug discovery and GPCR research get examined seriously. With Dr. Kanakin in the room, not content to consume, a place to think. All the links are in the show notes. And now, back to the conversation.
So wonderful. I think to your point, a lot of us thought about med school, and a lot of us realized you need to have a special, I want to say, brain setting in order to be able to go through the training and then actually do the work. I absolutely agree with you. I have the same issue with the sight of blood.
Yeah, it's quite something.
Exactly. No, I absolutely agree. I'd be curious to hear more. We don't not often enough have computational scientists, GPCR scientists on the podcast. So I'd be curious to hear about your perspective on, studying GPCRs from a computational standpoint, and how do you translate that information into the lab, or vice versa?
Well, it's I think as computational scientists, whatever the targets, whatever the field, you're very dependent on actually experimental work. You don't generate the data yourself. What you do is you're gonna do predictions. You're going to make predictions that are obtained through a predictor that was trained on experimental data. So, and we used to say, we say garbage in, garbage out. So if your experimental data you're basing yourself on is of poor quality or not well annotated or not well cleaned, well, then your computational work is going to be useless. So I think it's important for us computational scientists, and especially in the field of GPCRs, we need data. And I can only encourage people in the lab to please put your data online, make it available, add as much annotations as you can, indicate if you the conditional, the experimental conditions you use to obtain that data. If, in case you introduce a mutation into your GPCR sequence before expressing it, please tell us, because our work will depend on your work, and it will only reach a certain quality if actually your work was of high quality. So, and they say data is a new gold. So I think, please put your data online, and not only positive data, not only successful experiments. Also, I know it's researchers are not very motivated to do that, but putting your let's say unsuccessful experiments online, your failed experiments online, is also super useful for computational biologists. And then if we use that data, develop a model and have some predictions, well, then again, it's only just predictions. So we need, again, experimental people to validate our work. So we really need to work together and have this feedback loop between experimental and computational work to have something that is useful. So then some people might say, why do you need computational work if at the end you keep relying on experimental work? Well, there I think computational work allows you to look at the data differently, from a different perspective. It allows you to look for patterns, and let's not forget that in 2024 the Nobel Prize for Chemistry was co-won by AlphaFold, or the team behind AlphaFold, which is a computational work, and I think we have all already used AlphaFold, and if you haven't tried it out yet, it's super cool. So it's this model, this AI model that allows you to predict the 3D structure of a protein just based on its sequence information. And this is the kind of models we dream of once developing as a computational scientist. And here again, let's not forget that AlphaFold is so successful because of decades of experimental work. For decades, people have been making their experimentally resolved 3D structures online. The PDB has been curating them and taking care of this database. And thanks to that high quality data, AlphaFold, that was good enough to win a Nobel Prize, could be developed. And now again, we see that a lot of experimental people use AlphaFold to, for example, get an idea of how their 3D, how their protein looks like, the 3D structure of their protein, and then thanks to these predictions of AlphaFold, they can, for example, successfully obtain experimentally the 3D structure of their protein, which they couldn't do before, or they can predict which protein their protein of interest is interacting with, so a bunch of new insights. So I think it's super exciting.
I think you make a good point. I apologize. I think there is a delay between the two of us. I didn't mean to cut you off. We will clean this up at editing.
No worries. Please go ahead.
So what I was going to say is that you make a good point between this connection between experimental and computational work, because one supports the other. And those two things that came to mind while you were telling, the story and how you were thinking about the complementarity between the two. Number one is the need to collaborate with people in the lab, as well as people in the lab to collaborate with people at the computer, which is very important. And number two, one thing that's very important to think of, at least that's something I try to think of, is that you can, as two hands in the lab, you can generate only a certain amount of data, whereas in the computational world, it can handle hundreds of times of whatever you can generate in a week or in a day in the lab. And the accumulation of that good quality data is what gives the computation the power.
Exactly. Yeah, I couldn't agree more about that. I think, for example, in my case, my work, the goal is to develop a pipeline that supports the design of hybrid GPCRs in the lab. And so I think my only goal is to give hints to the people in the lab to know where they should start their experiments. And so yeah, I think it's super exciting that you can work together. And I think actually I would recommend everyone, both from the computational side and the experimental side, to find a collaborator from like the other side. So as a computational scientist, I'm very happy I've found some experimental collaborators, because it's a very fun it's very fun to work together, because I think people in the lab are wizards, and they think, I have the feeling at least, that they think we are wizards. And so you kind of like motivate each other by just like working together. So they would like us to design something, to predict something. So I get motivated because I know someone is waiting for my results. I'm generating these results. Then we have a meeting. I share the results. Of course, I'm super excited because the result. Then they go into the lab. They generate results. Then we have a meeting again, and it's very dynamic. It's very I think sometimes in academia, and especially maybe as a PhD student, you can feel a bit isolated and in a bubble. And so if you can find a collaborator that is from the other side, so again, for example, an experimental collaborator, if you are yourself a computational scientist, I think it's very fun. It makes your daily job way more interactive.
I agree. I absolutely agree. I think it's very important. And I think when it comes to, finishing a PhD or doing a degree like you're just doing, it's about learning the scientific skills, but also learning those communication skills, because scientists in the lab don't speak the same language as scientists at the desk in front of the computer. So I'd be curious if you have any, let's say, fun stories about how you learned to communicate with the bench scientists on some data. I'm sure there are stories there, and I'm happy to share some of mine as well.
I think it's a bunch of misunderstandings, the fun stories. Nothing is on purpose. We both didn't do it on purpose, but at the end, you're talking for like 10 minutes about something, and then you actually realize that the way that what you thought I was talking about is actually something completely different than like what I was actually talking about. So sometimes we use a different word to describe something, but that definition of that word is completely different for each other. And also I think we sometimes don't realize what is important for the other one. So for example, for us, it's super important. We know exactly which sequence we are working with. So if you introduce any mutations, if you have cut the C terminal, we need to know everything about that. And for over a week, I was working on a protein, and I was convinced I had to work with the canonical sequence, but actually they were working with the isoform. And to them, it was just a detail, but for us, it's a huge difference. So I think you learn along the way. You learn to okay, what is important to them. What should I definitely mention? And then also maybe sometimes just ask, like, are we on the same page here? Did you also understand this did you interpret the results in the same way? I think it's all about communication. And I think it's fine that we don't talk the same language. We just need to translate to each other what is important. So we don't have to be to explain every single detail of what we do, and so translate everything, because I think that's too complicated. You cannot expect from computational scientists to understand everything from experimental scientists and the other way around. But just try to figure out what is important for each other and make sure to translate that. And sometimes just take your time. Maybe you need to re-explain basics or things that you think are basics, but that might not be for the other one. And so it's also good to brush off your basic knowledge and explain that to your collaborators.
I agree. And you made a great point about not having to learn the other team's language, but it's finding that common ground. And the other important thing that you said, which I really like, is always checking, are we on the same page? And I think that's very important because it saves you not minutes. I think it can save you weeks.
Oh, yes.
And I think I agree with you on the importance of using the same sequence. To me, it's not if it's an isoform or if it's a natural, that's a big difference there. So
yeah, but I can really not blame the collaborator for that. I think for them it was not. I mean, it was obvious. Of course, we were studying that isoform, but it's just they forgot to communicate that, and I really don't blame them for that. I think I should be blamed that I know how important it is for us, and I didn't double check. So I think it's all about learning, and I guess at the end of my PhD or I will get better at that, but it's part of the learning process.
I think it's, to your point, I don't think it's about placing blame. It's about defining how do we work together and having that checklist of what do I need to ask the people in the lab and what do I need to give the people in the lab, and vice versa. What do they need to give me and ask me so that we're all on the same page? And I think that's where it becomes very important.
Yes, definitely. And I think there are a couple of I've realized that, for example, after every meeting, sending notes of the minutes of the meeting can be super useful to for example, check if we're on the same page, and rephrase it in my own words. I think also it's important to make sure that that we are both getting something out of the collaboration. I think the best way to keep a collaboration going is to make sure that, okay, this is where we are aiming for, and you are going to get this, and I'm going to get that. And I think if you have that, then it's going to be a successful collaboration. Unfortunately, I've seen, some collaboration, and they start on, everybody's very excited, but then you realize step by step that actually the other one has no incentive to really go 100% for it, and then you just lose your time. So again, I guess it's like a relationship. You just have to communicate as much as possible.
I agree. It's absolutely, I could not agree more. This is exactly how I see everything when it comes to human relationships, especially relationships at work. Because to your point, you have to write it down. You have to make it sure that everybody's happy with who's getting what, who's doing what. And it has to be mutually agreed upon, because I feel like life is too short to be working with people that you don't necessarily like to work with.
Yes.
Number one. Number two, we have better things to do than to work on things we're not excited about.
Yes, exactly.
So no, and taking notes, a little trick I use, I have, and I will not share this while we're recording, I'll share it afterwards. I use a system that allows me to record my meetings. And after that, it sends an email with the video recording and a summary to me and to the person I had a meeting with. And so I actually have a ChatGPT prompt that allows me to extract the action items. And I document that and send it in an email with a link to the recording. This way, if anything goes wrong, you can go back to the recording and say, no, you're on camera, you said this. And it's totally free. And it's not about blame. It's about figuring out what are the best ways for us to collaborate and to move forward towards our shared goal.
Yes, that sounds very useful. Again, a cool trick of AI.
Yes, I'll share with you the name of the one I use. It's a totally free version. I'd be
Cool, thanks.
before we continue, I'd be interested to, of course, I'd be interested to hear more about your PhD project. You kind of alluded in the beginning how you thought you were starting your project, but I'd love to hear more about what was the work about, and what are the, take home messages? What can we learn from the work that you've been doing?
Okay, so the final goal is to develop a computational pipeline that help people in the lab to design chimeric, some people call it hybrid GPCRs. So what maybe first I should define what hybrid or chimeric GPCRs are. So a hybrid GPCR is a construct where you basically gonna take a well-characterized GPCR. So a GPCR, for example, which ligands it binds, what is its signaling pathway. So, for example, beta-2 adrenergic receptor. And then you have another GPCR that is very poorly characterized. For example, orphan GPCR. And so the idea of a chimeric design is that you're gonna swap the loops. I especially focus on the intracellular loops of my well-characterized GPCR with the equivalent loops of my not well-characterized GPCR. So at the end, you obtain a hybrid, so a combination of two GPCRs within one GPCR. And then you can, for example, in the case of an beta-2 adrenergic receptor that will be at the extracellular side and provide also the transmembrane regions of your construct, and you will have your not well-characterized GPCR at the intracellular loops. You can put the ligands, well-characterized, well-defined ligands of your well-characterized GPCR. But now the signaling pathway you're gonna measure, you're gonna observe, is not gonna be the signaling pathway of beta-2 adrenergic receptor, but it's gonna be the one of your orphan, for example. Something you could not observed before because you had no idea how you could even just activate your orphan GPCR because the ligand of that GPCR is not defined. So the design of chimeric GPCRs, the concept of designing chimeric or hybrid GPCR, something that has been people have done it in the lab for decades, and it has already shown that in many cases actually that it can really help you to gain valuable insights into not well characterized GPCR. So insights at the for example, to characterize the signaling pathway, but also to understand which are the structural determinants for a particular to enable the binding with a particular G protein, and as such observe a particular signaling profile, but also to obtain, for example, the experimental 3D structure of a GPCR of interest. So these chimeric constructs can help you to stabilize your GPCR and so more easily obtain resolve the 3D structure. So where I come in here is that actually we also realize that even though chimeric GPCRs can be super useful, it's very difficult to design them. And it's not very, it's quite unclear at this point, like why it's difficult to design them, because for some combination of GPCRs that's like two GPCRs are very similar, you would think it's easy to design a chimera. Well, actually, it doesn't work. And then for some combinations where the two GPCRs are super different, even for like two GPCRs from different classes, well, then it just works. So my goal here is to help. So we have this computational framework where we're going to actually make a lot of possible candidates of these hybrids, and then we're going to test in silico, so on the computer, we're going to test, okay, how likely is it that this design is successful, is functional, and so we're going to rank all these candidates, and then we provide this ranking to the people in the lab, and then they can say, okay, I can test the top 5, and then hopefully it will enable them to answer some biological questions, and so they don't have to lose time trying a bunch of constructs without actually some rational design methodology. And so the experiment, like the work we do with the experimental group now, is actually to make some serotonin chimeric GPCRs, and through these chimeric constructs, we can now better understand or we try to better characterize the beta arrestin signaling pathway of a specific serotonin GPCR.
It's very, very interesting because, to your point, being able to try and designing these proteins in silico and then testing them and scoring and ranking them is so much, and I'm going to use air quotes, I don't like to do that, but so much easier than actually trial and error in the lab. And it allows you to get answers faster.
Yeah, exactly. At the end, I like to see my work just as I aim to just develop a tool that make other people's life easier, or at least their work life easier. And I think it's pretty nice because then you also know that if your project is successful, it will help others, and I think it's it's a nice incentive.
It is. I was thinking about, the orphan GPCRs, and I think it'll help the example you had with the beta-adrenergic receptor at the top, having the transmembrane domains, and then substituting the loops from the intracellular side with, for example, an orphan, then it'll allow potentially to figure out which G protein or what signaling does it activate. It still doesn't answer the question of what is its natural ligand. That being said, being able to have a readout and then do a screening to look for natural ligands is a beautiful way to de-orphanize.
Exactly. And you can actually also do it the other way around, make constructs the other way around, where you're going to put your orphan GPCR at the extracellular sites, and then, for example, your beta-2 adrenergic receptor at the intracellular sites. And then you can also, like, for example, do screening assays. And again, now you know the signaling profile of beta-2 adrenergic receptors, so you know that you found the right ligands. You found a binding partner for your orphan GPCR because you can you observe a response that is typical of beta-2 adrenergic receptor. So you can make all kind of constructs depending on what you're looking for, and I think it's just this idea of using knowledge you already have from this well-characterized GPCR to better understand and gain knowledge about another GPCR that is not well-characterized. I think it's a nice concept.
I think so too. And to your point, it's I'm not saying it's the same thing, but Kofi Therapeutics has this, I think, trademarked methodology where they actually have these chimeras that allows them to activate the same pathway or test multiple ligands with something that's already specifically primed towards a specific pathway.
Exactly. Yes, exactly. And so maybe I forgot to mention, but so what is I actually comes back to the beginning of our discussion. What was super important for my work to develop this. I'm still wrapping up my pipeline, but it's getting there. What was super important was the data again. And so I think a problem we have a bit in the field of chimeric GPCRs, and so maybe it extends to the field of GPCRs in general, is that it's not well defined. So some people describe these constructs as hybrids, others as chimeric constructs. Some just say we made a construct, and so it was very difficult at the beginning of my PhD to gather all this data, collect all this data, and then I also realized that in some paper they were discussing a particular design, and they observed that they could reach a 50% functionality compared to the wild type. And then in another paper where they made exactly the same construct, they would see that they could reach 100% activation, like the same activation profile, signaling profile they would observe for the wild type. And so then I was like, okay, but so which one is the best? I mean, for same construct, you would obtain two different results. So it means that perhaps the difference is in the assay they used or the experimental conditions. So I think it's there that you realize it's so much more complex than it looks like on paper. And so, to be honest, it took me over a year to gather all this data. And now we have actually made it publicly available, this data in a database. called GPCR ChimeraDB. And so it's all there now, and you can, it's a very interactive database where you can look at this, so previously designed chimeras, and also you can connect that data to the two parents, so the two GPCRs involved in that design, and you can look at the construct from a sequence, structure, or biophysical perspective. So it's pretty cool. I would encourage everyone to go check it out.
We will add it to the podcast page so people can find it very easily. I actually have it bookmarked. I saw your post, your initial post on LinkedIn, and that's when I thought, oh, this is actually really cool work.
Nice, thanks.
To your point, when it comes to, you were mentioning, same construct, same readout, two different papers, different activities, and this is every GPCR biologist's problem, where you're like, what just happened? And to your point, it might be a different type of kit or different type of assay, but I think it is the pain point.
Yes.
When it comes to cleaning out and normalizing data in order to use that data in a computational context.
Yes, and there again, if we are all super transparent about what we exactly did, it would make it a bit easier to gather this data and compare it. But on the other hand, it's not just about that. Sometimes we don't realize that actually it might be important to someone else to mention it. And so here again comes the point that we need to understand what is important for each other in order to make most out of it. For both the experimentalists that generated the data, which I'm sure would be super excited to share the data with me and discuss the data with me, but also for the computational scientist that is super excited when they finally find some work that is super well annotated. So I really think that now computational work is getting more and more sexy, let's say, and we see a lot of work with, for example, now protein design of or the design of mini binders, so small proteins. And I think the work of David Baker and his group has been leading, has had a large impact on that. But share your data and annotate it, please.
I agree. As you were explaining the fact that, different papers called the chimeras in a different manner, the first thought I had, I think this is a good topic for IUFAR in order to put a small meeting together, a small group together to standardize how we call these constructs in order to make sure that we use the same nomenclature. I think it's time to do that.
Yes, I agree.
It's time to do that.
But who knows, maybe if it gets easier to design chimeras in the lab, thanks to computational tools, then maybe people will start designing them more often, publish more often about them, and then it will even become more important to define that nomenclature.
Yeah. Maybe we need to your point, reach that critical mass in order to say, yes, we have to now standardize it and give it enough name. But hopefully your database and all the work that you put into your PhD work is going to pay off and lead to, at a minimum, and obviously I'm completely downplaying the whole importance of your work, but at least people will think about, we need to standardize this.
Yes.
To make every computational chemist happier, but also every biologist happier.
Exactly. And if it's just that, I'm already super happy. I think that would already be very nice.
It would be actually a fantastic thing. Me, who likes to organize things and classify and checklist things, and I think you're a little bit like me as well. I think that would make everybody happy.
Yes, definitely.
All right, Charlotte. Top 3 aha moments that shaped your PhD or your scientific career so far.
I think I don't think I had it yet, at least, but I've been thinking about this, and actually I think for everyone that works with GPCRs, they might not have a aha moment yet, but I'm sure they had a lot of gotcha moments, because GPCRs, they're so fascinating, and they really keep you on your toes. Every time you think you might start understanding how they work, they're like, no. Gotcha. You're not gonna figure it out yet. Have you considered this? Did you look into that? And so I think it's why it's super exciting to work on GPCRs. And like I was saying before, it's a pure coincidence. I work on GPCRs, but I'm actually super happy at the end to be part of the GPCR community and the GPCR research field, because it's such an interesting target, and there's so many ways to look at the target, and it's also a very active field, and I think it makes it quite motivating that you see that every now and then there's a new paper that comes out and like makes you rethink your work, and I think it makes it very interesting.
I agree. So actually, thank you for your gotcha moment, because I actually edited the episode outline, and now it says top 3 aha or gotcha moments. Because to your point, we have a lot of gotcha moments.
Yes, I actually hope so, because otherwise I would feel like I'm the only one not getting it.
No, it's not you. But I think maybe to your point, the gotcha moments lead to the aha moments.
Yes, true. It's part of the process, part of the journey, I guess.
I think so too. Best advice for a scientist entering the computational side of the GPCR field?
I would say maybe 2 things. First, but that's, I think, for any young scientist, dare to ask. They say a no you already have, a yes you might get, and I know it's super. I try to live by this, and I think it's sometimes very difficult, but because you have to put yourself on the spot and like ask. But I've realized that actually, like in most cases, people are happy to help, and so of course, if you need to phrase your question wisely, because people are more likely to help you if they get something out of it. But in most cases, people will just say yes. So just dare to ask. Dare to ask your supervisor to do a secondment during your research project. Dare to ask if you can go to a particular conference, because if you really want to go, then I'm sure your supervisor will consider that. So I would say that, dare to ask. And then second, and that maybe applies more to computational scientists, is please don't stay in your bubble. I think we can get so easily isolated and maybe lose some perspective about our research. And then I think then it's super easy to get demotivated, because you lost maybe the broader picture. You forgot that actually you're working on this tool because at the end you wanted to help people in the lab obtain better results. And so if you don't go to conferences, if you don't go to workshops, then you might stay in your bubble. And if you're lucky, you can maybe talk with your colleagues, but it's very likely that they're doing the exact same research as you're doing. And so, get out there to get a different perspective on your work.
Agree. And maybe I want to add to that. I feel like whenever I used to do data analysis at my computer, you have your cup of coffee, your cup of tea, whatever you want, and you get comfortable, and then you go into that bubble.
Yes.
And to your point, you have to get out of the bubble.
Yes, exactly. And I mean, nobody likes to go out of their comfort zone. But I really think that at the end, it's what you're going to get, like, or at least for me, for example, it's that's what I get the most energy out. After a workshop, after a full week of like exchanging ideas, discussing with like fellow PhD students that are going through the exact same things as you, I come home and I'm like, okay, I'm super motivated now. I'm really excited to like get back into it. So yeah, go for it.
No, I absolutely feel you. I think it exactly. And to your point, if you don't ask, it will always be no. And well, not necessarily about your supervisor, but in general, if you don't ask, if you need something, you only need one yes.
Yes.
You might get 37 no's, but you need one yes.
Yes, exactly. And then maybe just about a supervisor, for those that are maybe considering starting a PhD, choose your supervisor wisely. I think it makes a huge difference. I'm super fortunate with my supervisor, and I think it at the end of the day, it's really gonna make the difference. So don't just go for, oh, I see a PhD project that is fully funded. Let's just jump right into it. Maybe double check with the people that are currently working in that lab, how the vibe is, how the working environment is, and then maybe based on that, you can then make a decision.
I agree. One last question. You may not have the answer to this, but what next after your PhD? Only if you're comfortable sharing.
Of course. To be honest, I'm not sure. I was hoping after my master's that a PhD will help me clear everything out. And actually, the only thing I know is that I would be super excited if I can continue working on GPCRs in one way or another. Because like I said, I have the feeling there's still so much to unravel, and I'm very excited to be part of that story. But then if it's going to be academia or industry, I think both have pros and cons, and I think both also require a specific set of skills. And in my eyes, I think both are interesting options. So we'll see. But in any case, I think doing a research project after my PhD, if it's going to be as a postdoc or in a company, I think it could be super interesting, because now I have this feeling I'm in my final year and I'm finally getting it. I'm not getting GPCRs, or at least not fully, but I'm getting how research should be done, or more or less like how I should organize my work, what need to be like the steps I should pay attention to. And I have the feeling at the beginning of your PhD, you lose a lot of time making mistakes, which is perfectly fine, but you lose a lot of time. And now I'm actually, I finally got it, but my PhD is ending. So I think it can be super interesting to start a new research project with all this experience in back of my mind, and of course I will make more mistakes. I'm 100% sure about that, but perhaps I will know better how to handle them and how to like learn from them, and everything can go maybe a bit faster. So research and GPCRs. I think if that's in my future, I will be very happy.
And to your point, I feel like when you finish your PhD, you get to a point where you're like, I have my own ideas. I learned from a lot of the mistakes that I made. And to your point, it's okay to make mistakes. And I felt the same way. I said to myself, bring it on.
Yes.
Like, throw something harder at me right now, because I've learned something and I want more.
Yes. You want to go for the next challenge.
Exactly. Fantastic. Charlotte, thank you so much for your time. I really enjoyed our conversation. Good luck on finishing up your PhD. I'm happy to offline, obviously, offer some ways to think about, what's next. But one piece of advice is, no matter what you do your homework, talk to people, exactly just as you said, and then go with your gut at the end of the day.